Alright, we're going to cover section 1.2, and before getting into what is written in the study guide, thought we look at this, the structure of normal tissue. Epithelial tissue has this sort of structure that I'm showing here, where you have a layer of epithelial cells, and the epithelial cells are on one side facing a cavity. Now, the epithelial cells are tethered to a basement membrane by membrane proteins that they have. So I'm just gonna draw some of these lines, and these are supposed to be proteins that are on the surface on this side of the epithelial cells that are binding into the basement membrane. On the other side of the basement membrane, you have stromal cells. Now, these are two different cell types, epithelial cells and the stromal cells. And all tissue is basically organized in this way. Now, the reason why this tissue or any tissue in a multicellular organism such as ours functions correctly to maintain life is that these cells are doing the correct things because they're surrounded by other cells and because of their environment that they're in. So if I remove one of these cells from this environment and put it into an artificial environment like a petri dish, they will start to behave differently. Their phenotype will change. They'll retain some of the characteristics they had while they were in the tissue, but they will lose some of the characteristics also. So what is this getting to? Each individual cell is an independent agent. It has been programmed to behave in a certain way in a certain environment. So for it to act correctly to maintain life in tissue, it has to be surrounded by the correct cell types and be bathed in the correct fluids with the correct proteins and signaling molecules and etc. factors that lead to it expressing the correct phenotype. Otherwise, if this environment that we're showing here changes, those cells' phenotype will also change. That will lead to the cells not behaving in the most optimal manner to maintain and support life. That the key to every biological problem must finally be sought in the cell, for every living organism is or at some time has been a cell. The cell is the smallest unit of life, and you are nothing but a collection of cells, each working independently, but yet in concert with each other because they're in the correct environment. So this whole idea of every single cell being an independent agent, but yet when they are together, they maintain life, and you have these amazing organisms that do amazing things, are able to do amazing things, makes the problem of cell biology very complex. But the one thing that is sort of in our favor in trying to figure out what is happening is that there are these fundamental mechanisms that all cells they have in common. So, for example, the one that most of you will know is just transcription and translation. All cells use transcription and translation to express their genetic code. So gene expression, the method by which it happens, is a common mechanism across all cells. And there are other common mechanisms that we will be learning about during this class. And we can just assume that these common mechanisms will be the same across all cell types. There might be slight differences, but those common mechanisms are going to be a constant. So one of the big things that students have trouble with in this class that I've noticed is the difference between a why and a how question. And we're not really interested in the why, but we're interested in the how. Some of the example of why questions: why does a cell respond to its surrounding environment? I mean, that answer as a why question is very simple, because to maintain life, it must respond to its surrounding environment. That's part of one of the definitions, one of the characteristics of life. A living thing will respond to its surrounding environment. But that in itself is not that interesting, but the how question is interesting. How does a cell respond to its surrounding environment? Because then you're getting into the molecular mechanisms that are happening in the cell, and that is what we're concerned with. So sometimes a why question is presented, but it's still a how question. And I have a couple of examples here of two questions. And these two questions, we're going to actually answer them in this class. The first one we're going to answer in section 1, and the second one we're going to answer in section 3. Looking at example 1. Cells utilize fatty acids at a much lower rate to meet their energy needs under anaerobic conditions than in aerobic conditions. Why is this the case? Why under anaerobic conditions does the cell not use fatty acids? And there's a molecular answer for that. Because fatty acids enter cellular respiration at the citric acid cycle, and the citric acid cycle requires oxygen. So if you're under anaerobic conditions, the citric acid cycle is not working. Because of that, fatty acids cannot be utilized for energy use. Okay, so moving on, a little bit more chemistry. Cells are 70% water. Life depends largely on chemical reactions that take place in an aqueous environment. Oxygen and nitrogen. Oxygen and nitrogen are electronegative atoms. All that means is that when they form covalent bonds with electropositive atoms, and the one electropositive atom that we're only interested in is hydrogen, and in the case of nitrogen, it can make 3 covalent bonds, 3 single bonds. When oxygen and nitrogen are single bonded, or they're bonding with electropositive atoms, such as hydrogen, the electrons that are in that covalent bond will spend more time around the electronegative atom. What will happen in the case of water molecule, the electron that hydrogen brings to the covalent bond will spend more time around the oxygen than it does around the hydrogen. So what will happen is that you'll have a partial positive charge on the hydrogens and a partial negative charge on the oxygen. The same thing for nitrogen. The electron that the hydrogens are bringing to these covalent bonds will spend more time around the nitrogen. So you have a partial negative charge on the nitrogen and partial positive charges on the hydrogen. And all that does is it creates a dipole moment. In both cases, what you'll have is you'll have partial negative charge on the top, a partial positive charge on the bottom. And this will allow for nitrogen and oxygen of other water molecules, for example, or other ammonia molecules to bind to other molecules such as this. So for example, water molecule will be able to form an electrostatic interaction with this water molecule. We'll just have it as these 3 little dots and note an electrostatic interaction. It's not a covalent bond, but it's an electrostatic interaction between negative and positive, because negative and positive are attracted to each other. Same thing with an ammonia molecule. I put an ammonia molecule here, and here's our electrostatic interaction, right? So this an oxygen-hydrogen single bond is a polar covalent bond. That means that when you have this interaction of this covalent bond formed between an electronegative atom and an electropositive atom, the covalent bond is going to be a polar covalent bond because the electrons in that bond are spending more time around the electronegative atom, in this case oxygen in the case of water. An oxygen-carbon double bond is also a polar covalent bond, so when we're looking at something like this, there's going to be a partial negative here and a partial positive here. And we already covered the nitrogen-hydrogen single bond. So carbon, hydrogen, nitrogen, and oxygen are the most common elements in living organisms, making up to 96% of an organism's weight. So functional groups. This is not something that I'm going to test you on. The name, like for example, the question where what does a hydroxyl group look like, and etc. I'm not going to test you on this, but I'm going to use this language, like a hydroxyl group or a carbonyl group. And I'd just like you to be aware of it so that if I use that language during the course, you know what I'm talking about. So, for example, a carbonyl group is just a carbon double bonded to an oxygen. A hydroxyl group is just simply an OH group. The carboxylic acid group is going to be this thing right here, a carbon double bonded to an oxygen and single bonded to another oxygen. And at pH 7, a carboxylic acid group is going to be have a negative charge on it. So here's our carboxylic acid group. But at pH 7, it's actually going to be this. The hydrogen will leave, and it will leave its electron behind. So it'll leave a full negative charge on the oxygen. Ester groups, which is just a carbon double bonded to an oxygen, single bonded to another oxygen, and that oxygen being single bonded to another carbon. The amines, where you have an NH2 group, but at pH 7 it'll be an NH3 group, so you have a full positive charge on this group. So typically this is the way that an amino group or amine group is written, is at pH 7. So here's my carbon, NH3 plus. So at pH 7 it's going to have a plus charge and 3 hydrogens, one extra single bond being made by this nitrogen. Amide groups, this is important for peptide bonds because peptide bonds are amide bonds. And we're not going to really deal with thiol groups, so this is not super important for us. Phosphates are always important, a phosphate group, because phosphorylation is a common theme. So for example, ATP is a triphosphate, so it has 3 of these phosphate groups attached to each other in a linear fashion, like it's being shown here. This is a diphosphate. And a phosphate group is going to be abbreviated with a P with a circle around it. And that's being shown right here. Okay, and we'll end this video at this point. Thank you. I'll talk to you guys later.